नजर आ रही है. So what can you see in the screen?
TSH.
It's a TSS, right? Okay. So TSS, today the third experiment we are going to learn, and the aim of the experiment is to determine TSS concentration in serum sample. The method we'll know about is the ELISA method, which is a sandwich ELISA based method. ELISA full form is enzyme, E for enzyme, L for linked, and I for immuno, S for sorbent, A for assay. Enzyme linked immunosorbent assay, where TSS means T for thyroid, S for stimulating, S for hormone. Thyroid is stimulating hormone. So I hope you have no any doubt in this. So this is the aim. We are going to we have experiment today, and the aim of the experiment is to determine TSS concentration in serum sample. Okay, and method is what we need to know is a ELISA, basically sandwich ELISA. So general information about the TSS. TSS is a glycoprotein. So it's a glycoprotein means a type of complex protein where proteins are conjugated with complexed with uh, glucose moieties or carbohydrate moieties. Okay. So this hormone is glycoprotein hormones, and this TSS is secreted by the anterior pituitary gland. Okay, basically the anterior part of the pituitary gland, and this TSS has a target cell, target organ, so it act on thyroid gland. Okay, so secreted from anterior pituitary gland, and through blood channel it goes to thyroid gland, and it act on the thyroid gland. uh, what is the role of this TSS? So it regulates the synthesis or release of T3 and T4 by thyroid gland. Okay, so this is the one of the important function of TSS, that it helps the regulation, either increase or decrease. So when there is a TSS secretion by the anterior pituitary gland, so it causes stimulation of synthesis or release of this T3 and T4 by thyroid gland. Okay, this is the important function. Uh, TSS has two subunits, namely alpha and beta. There are two subunits in the this this hormone is a dimeric hormone. Okay, there are two monomers, two subunits. One is alpha and one beta. So its alpha subunit is similar to, okay, alpha subunits, which is has a amino acid sequence which is similar to LH, FSH, and what we say. Uh, there is some problem with the text. Let me change the color of this. Okay, now it's looking. So what we say that the two alpha units and one beta subunits. So it's a two subunits. It has one alpha and beta, and alpha subunits is similar to what we say LH, FS, SCG. SCG is the human chorionic gonadotropin hormone, and it's a glycoprotein hormone. Okay, so there is some mistake here. Let me delete this. So the three hormones, alpha subunits are similar. LH,
Sir, your
FSH,
voice
and
is not
human
clear.
chorionic gonadotropin. My voice is not clear.
No, sir, it cutting.
Sorry.
सर बीच-बीच में से कट हो
Is
रही
it
है.
बीच बीच में कट हो रही है? Okay. Uh, are you able to see the screen?
Yes, sir. Now it's clear. अभी क्लियर हो, जब ज्यादा आप बोल रहे हो तो सबसे
It's
बीच-बीच
clear.
में अपने आप ही कट हो जा रही है.
Okay, okay. So okay, let's start from the beginning here.
सर फ्रॉम देयर
That
अल्फा सब यूनिट से स्टार्ट कर दो एक बार.
okay, okay. So TSS has two subunits. Okay. So when we study the structure of TSS, it has two subunits. Okay, it has two monomer units. One is alpha and another is a beta. Alpha subunit is similar to another hormone like LH. LH is a luteinizing hormone. FSH is a follicle stimulating hormone, and HCG is a human chorionic gonadotropin hormone. So this alpha subunits of TSS is also similar to the alpha subunits of LH, alpha subunits of FSH, and alpha subunits of human chorionic gonadotropin. Beta subunit is TSS specific. So if you see the structure of beta subunits, it is unique to the TSS only. It is not similar to the what the beta subunit found in LH, FSH, and human chorionic gonadotropin. Right? So alpha subunits are similar, whereas beta subunits are unique and it's very specific to TSS. I hope you have understand about the two subunits of TSS. The clinical significance. TSH measurements help in the diagnosis of thyroid disorder. This why do we measure this TSH? Because it helps in what diagnosis of which kind of disorder? Yes, thyroid disorder. Because TSH is produced in anterior pituitary, which has a target to bind on thyroid receptor. Okay, thyroid receptor present on thyroid gland. So when the thyroid gland is either abnormal or normal, so it is we can know by the measurement of TSS. Increased serum TSS is an early and sensitive indicator of thyroid uh, function. So if the TSS is increased, so in such condition there is a decrease in thyroid function, also known as hypothyroidism. Hypothyroidism is a condition where hypo means less, thyroidism means the condition where there is a disorder of thyroid. So hypothyroidism is a decreased thyroid function. Okay, so in decreased thyroid function, TSS level is always higher side. When there is a decrease level of TSS, what it indicates? It indicates that there is a hyperthyroidism, means uh, there is a decreased thyroid function level condition. And this is a grave disease. Okay, so it is a grave disease, an autoimmune disorder. Okay, so in this autoimmune disorder, the grave disease, there is a increase of the thyroid
So your voice not audible.
right? My voice is not audible.
Yes,
So can
sir.
you explain again
Yeah.
from increased serum?
This TSS is sometimes increased, sometimes decreased. Okay. So if the value of TSS is increased, so it suggests that there is decreased thyroid function. Okay. Also, we can consider this decreased thyroid function as a hypothyroidism. Okay. When there is a decreased level of TSS in the blood, so it indicates hyperthyroidism, means the increased thyroid function. Hyperthyroidism means increased function of the thyroid gland. And this is the example of the hyperthyroidism, that is a disease which is an autoimmune disorder. Right? Is it clear?
Yes, sir.
So working principle, let's know about how this TSS measurement based on ELISA, how it works. So here we have been given the samples containing TSS. So we have a serum sample with where we need to know what is the concentration of TSS in the sample. So this is given. Then there's anti-TSH HRP conjugate. So this is the antibody which is labeled with HRP, horseradish peroxidase. So this is antibody which is labeled TSS antibodies. And another thing which has been provided is a wells, micro wells, which is coated with the anti-TSS. Okay. So antibody which is specific to TSS are coated to the TSS. Okay, so we have three things specifically which has been provided. One serum sample, one antibody coated antibodies, and another is antibodies which are coated to the microvales. Okay, which is immobilized antibodies, which is not coated with any enzyme. So the TSS in the sample forms a sandwich between the two specific antibodies. So TSS in the sample, what happens? what happens? It is sandwich. It is sandwich between, okay, two specific antibodies. One is a NTTSS HRP conjugate, another is a cold Wells protein with the NTTSS. So this antibodies which is in the Wells, and other antibodies is the enzyme level antibodies. So this TSS is sandwiched between these two antibodies. Okay. So unbound TSS and HRP conjugate are washed out. So after when the binding takes place, when sandwich formation takes place, so the next we need to wash off the this unbound, which is free TSS and free HRP conjugate. Okay. Upon the addition of substrate, that when we do washing procedure, then when we just add the substrate, which is specific to the HRP enzyme or reduced peroxidase. So this according this substrate acts on the HRP, and then color is developed. Okay, so upon the addition of substrate, color is developed, and the intensity of color is proportional to the concentration of TSS in sample. So more the color intensity, more will be the TSS sample, TSS in the concentration in the sample. Standard curve is relating color intensity to the concentration of TSS. So we can prepare the standard curve by just relating to the color intensity, okay, color versus in concentration of TSS. So what is the color absorbance and what is the concentration of that particular standard? So because based on that we can prepare a standard curve, okay, and from this standard curve we can be able to detect the TSS in the unknown serum sample. So this is what from this we are able to detect the value of TSS in the given sample. So requirements: microwells coated with NTTSS. These are the requirements. TSS standard, 7 vials of TSS standard with a different concentration of TSS. TSS conjugate reagent, which is the enzyme level antibody. Then TMB, which is a tetramethyl benzidine substrate. Okay, and we have a stop solution. We have a 20x wash concentrate, means the wash buffer. So these are the requirements we need to provide. I hope you have the information about what is the role of microwells, what is the role of TSS standard, what is the role of TSS conjugate reagent. You should know the function of this. Okay, you can ask me if you have the doubt for related to this, the function of this, then you can ask me at the end. Materials required but not provided. What are the materials required? Distilled water and deionized water. Okay, this either distilled or deionized water we need to arrange. We need to arrange pipettes, what we call precision pipette, disposable pipette tips. So tips is also need to be provided. ELISA reader also need to be provided, where we can record the readings of the test standard at 450 nanometer. And also we require absorbent paper or paper towel or paper towel. We need to prepare a graph paper, a standard curve graph paper is arranged, and we need to arrange the timer also. We can see the timer where incubation period is required for active time. So these are the requirements. What is the ELISA plate reader? This is the micro plate with holders, and this is the micro pipette or precision pipette. This is the tips, and this is the tip box. This is how we need to assemble this micro wells in the holder. Okay, so one set of the strip of this micro wells contain 8 wells, 1, 2, 3, 4, 5, 6, 7, 8 wells, and there are 12 rows, so 8 12 are 96. So this holder can hold around 96 wells in the holder. Specimen collection and preparation. That what specimen we require serum, do not use hemolytic, ectic, or epimic specimen. Please note, sample containing sodium azide should not be used. So this avoid it, that sample should not be added with the sodium azide, and this sodium azide inhibit HRP enzyme activity. So Odds radius peroxide is enzyme activity inhibited by sodium azide. So it is suggested not to use a sample containing sodium azide. Specimens can be stored for up to 5 days at 2 degrees Celsius, or we can store sample up to 1 month at minus 20 degrees Celsius. So this is the specimen extent very important for the specimen type. Now, dilution of 20x wax buffer. So, as per the what the instruction has been given, prepare 1x wax buffer solution by adding a 10 of 2% water or the water. Also, this way, this is what the given instruction from E. We have 25 so and mix 5 ml into 75 ml distilled water and mix water. In store at room temperature, procedure, the general remarks and information before I got procedure. That we need to bring all the reagents and specimen to room temperature before use. So very important that starting the experiment, all the steps, all the reagents, all the specimens should be brought to the room temperature. All reagents must be mixed without foaming. So ensure that reagents are properly mixed, because on storage some of the ingredients settle down. So we need to mix it properly and ensure that there is no foam formation. The steps should be completed without interruption. So when we start the procedure, all the steps should be completed without interruption. There should not be any delay in any of the steps. Always use new disposable tips for each standard, control, and sample. This is what is suggested, that to avoid the chemicals. So you need to always use the new tips, okay, new disposable tips. So let's know about this. This is how we need to prepare. You need to add the reagents in each of the sample using the multi-channel pipette, or you can use single channel pipette one by one when you it. So it depends upon the facilities that you have. It is suggested to use multi-channel pipette, which avoid the delay in the procedure. So let's know the procedure now. Place the desired number of microvales in the holder. That whatever the microvales that requires. So all the microvales, that the number that you have required, so you have to place on the holder. Now pipette 50 microliter of TSL standard. So we have 7 vials of standard with a different concentration. So all 7 standard vials should be added with the 50 microliter of the standard. So different concentration of TSL standard. So what we require, how much we require? We require 50 microliter. We require control. We require specimen. So all the designated wells should be added with the 50 microliter of this standard, 50 microliter of control, and 50 microliter of the specimen, that is a serum. Add 100 microliter of ready-to-use conjugate reagent to all wells. So conjugate reagents is an enzyme-labeled antibodies. Okay, so we need to add 100 microliter in the wells. Then after that, just need to shake for 10 to 30 seconds. This is very, very important steps, because this is what helps to ensure the proper mixing of this conjugate with the what there's respective standard, respective control, or respective specimen. So mixing is proper. You cannot do process properly. Okay? So need to mix this standard in minutes sufficient for this procedure. Next procedure is the report of it, and after that we need to cover that it with aluminum, and we can incubate for 60 minutes at room temperature. So to remove liquid from the wells, then when the incubation is complete, so you need to remove all the liquids which are present in the wells. So you have to discard all the liquids of the well, each wells. Wash well 3 times with one buffer. Okay, so each well should be washed 3 times using 1x wash buffer. Blot on absorbent paper transmit. Blot on means just remove all the liquid which are present in the well, so that it can be absorbed out. So blotting is called as a blotting, which absorb the all the liquids of each wells. 800 microliter of TMB substrate. So TMB substrate is a benzene, tetramethyl substrate which contains H2O2 also. So we need 800 microliter, and when we get TMB substrate in wells, then we need to see the color formation. So that should be the incubation for 15 minutes at room temperature, and after that we can see color formation, and then we need to add 50 microliter of stop solution to all this. So when 50 stop solution is added, then within the 15 minutes we need to so after adding 50 microliter stop solution, shake plate gently to make the solution, and at 450 nanometer within 15 minutes after adding the stop solution. This is what it is advised, that when we add 50 microliter of stop solution, so just to ensure that there is a proper mixing, and then after that we can take the OD at 450 nanometer in the ELISA reader. Okay, we can prepare the standard curve also. Standard curve, for standard curve preparation, this is the guidelines, this is the instruction you need to follow. Pick TSS standard value on each standard vial. So you have the 7 vials, and each of the vial the standard concentration is mentioned. So you need to check that what is the concentration of the TSS standard on each of the standard vial. To construct the standard curve, we need to plot the absorbance of TSS standard in vertical axis, that is a y-axis, and the standard concentration in the x-axis, that is horizontal axis on a linear graph paper. Okay, so the absorbance that we have taken for each standard, so we have to record accordingly, and you have to plot the graph absorbance versus standard. Okay, so absorbance should be plotted at the y-axis, and the standard concentration should be plotted at the x-axis. Okay, the best curve through the points, whatever the points we get, because there is always we have to join all the points, and we can get a straight line curve. Okay, so read the absorbance for control and each unknown sample from the curve. So once this standard curve is made, so we can be able to read the absorbance of control and unknown sample, and then after that we can get the value of this control and the unknown sample, and be able to give the values, and you can be able to report the value of TSS in the given sample. Report value of each control or unknown sample. This is what I know as sample, like A can be like standard 1, 2, 3, 4, 6. Control, but we consider it as a sample. So accordingly we can test it. Example of so this is the absorbance at the y-axis and concentration at the x-axis, and this is what we can get the linear graph. Under 1, concentration is 0, minus 7 per ml. This is of you have understand. Normally in the cases in male, basically in adult, is 2 microinternational per ml. Whereas in newborn, the values are very higher, approximately up to 39 microinternational per ml. Again, 2 on the low side, 1.7 to 9 micro international per ml, and 21 weeks to 20 years, value is to 6 micro international per ml. Okay, and in adult, what we consider in all body, the TSS value is 2.2 microgram per ml. Okay, so hope you have no questions, or if you have any questions, you can just drop me. Okay, we have already got so many questions over here. No question is there. Chatting box, there is no question, but the message is from Priya. Voice is breakable. Voice is not clear. Voice is not clear. Pradeep, yes sir. Any question, please? Yeah, it's time for question.